Welcome back, ladies and gentlemen, and thank you for joining the 80th Annual Academic Sessions of the Gall Medical Association. Proceeding to the 11th session of our program, we have the third symposium on a timely topic, COVID-19, Facing Emerging Challenges with Confidence. To chair the session, I cordially invite the co-chairpersons, Professor T.P. Veeraratne, Senior Professor in Medicine, Faculty of Medicine, University of Ruhuna, and Dr. Krishantha Jayasekara, Consultant Physician, Teaching Hospital, Karapitiya, to the head table.
Good afternoon. Welcome to the third symposium in the annual academic session. My co-chair, Dr. Krishantha Jayasekara, we'll be having three eminent speakers in this symposium named Facing the Emerging Challenges with Confidence. The speakers are Professor Suranjit Senaviratne, Professor and Consultant in Clinical Immunology and Allergy, Dr. Chandima Jeevandra, Director, Allergy and Immunology and Cell Biology Unit, University of Sri Jawadhanapura, Dr. Ananda Wijayakrama, Consultant Physician at the Infectious Disease Hospital, Colombo. Let me first introduce you the ground rules of this largely a virtual symposium. Each speaker will have 25 minutes, and the questions are entertained at the end of all three presentations and not at the end of each presentation. Audience, please type your questions in the Q&A box in the Zoom screen at the lower border, and do not ask questions orally or type in the chat box. It has to be in the Q&A box. I'll hand over the introduction of first speaker to my co-chair, Dr. Krishantha.
The first talk is going to be on COVID-19 vaccines, current global perspective. And let me warmly welcome Professor Suranjit Senaviratne, Professor and Consultant in Clinical Immunology and Allergy, Institute of Immunity and Transplantation and Health Services Laboratories, London, UK. Over to you, Professor Senaviratne.
Thank you. I'll give you a current global perspective on the COVID-19 vaccines as at the present time. All of us would know that this has caused massive devastation across the world, with the number of cases over 220 million, together with 7 million cases in the UK and many cases in the US and India. This figure will show you the SARS-CoV-2, a schematic of the SARS-CoV-2 virus. And why I put this is to show you the spike proteins on the surface that binds to the cell. The virus enters the cell, multiplies, and then spreads from cell to cell. Now, the vaccines have been designed to target the spike protein. And secondly, as the virus keeps on multiplying, variations can occur or mutations can occur, and therefore viral variants will have arisen, and this is cause and effect around the world. So vaccinations have been a major pillar when it comes to the control and of SARS-CoV-2 infection and preventing it and trying to get control over the pandemic. So 8 to 10 vaccines have been approved so far, but this slide shows you that there are some vaccines that were abandoned. That's not every vaccine that was developed was a success and was approved. So several vaccines were abandoned, especially in Australia, etc. And the rigorous process was followed with regards to doing the clinical trials and authorizing or approving these vaccines. Over 5 billion doses have been administered in the world so far. And you can see the schematic, this graph from May showing the countries that were vaccinated at high rate at that time. And then this was a more recent figure, again showing the countries that have very, very high second dose vaccination rates. Now, immediately you can see from this that some of the richer countries were at the top, and some of the countries were not even having a single dose of vaccine being given to its population. And this is not good because it means that it's not an equitable distribution and it has an impact on the variant distribution, etc. The summary of my talk would be quickly going through some immune aspects of the infection. Just 2 or 3 slides on the variants because that has been touched already. And then I will focus on the vaccines and what is new and what is current at the moment with regards to COVID-19 vaccines. If you start with the immune system, you have viruses, bacteria, and fungi that try to attack our body, the danger signals. You have the first part of the immune system, the second part of the immune system, antibodies, white cells, lymphocytes, and cytokines, which come into play to get rid of these. Similarly, in the SARS-CoV-2 infection, cytokines are just chemicals that help the immune cells to talk to each other. And if the cytokines are dysregulated, then you find that hypercytokine syndromes occur, and then issues occur with regards to fluid leakage, etc. What are the changes, the immune changes that occur in COVID? Many changes occur. Some cytokines are increased many-fold, such as IL-6, IL-1, etc. Some cells are reduced, some cells are decreased. And I won't go into great detail because I've spoken about this in previous talks, etc. But a lot of immune changes occur. And there is immune dysregulation. So the virus comes and multiplies, and then it leads to an immune dysregulatory state. And what's the importance of knowing this? By knowing this factor, we can target certain treatment options to control this process. So these are some of the treatments that we use, steroids, etc. And the important guidelines to be followed is continue the regular medications. And if it's an asthma patient, you continue the inhalers. You don't start it anew, but you continue it. Paracetamol is okay, not NSAIDs. You don't start aspirin newly, but if you're already on aspirin, you continue it. The important aspect to remember is steroids are, such as dexamethasone, is fine, but not given at the wrong time. It has to be given at the correct time, because if given too early and too high dose, it can cause issues. You do not give antibiotics unnecessarily, because this is a viral infection, not a bacterial. If you have a secondary bacterial infection, definitely, but you do not give it too early and unnecessarily. So these are some sort of guidance with regards to treatment, which you can get by studying the pathogenesis, and that is also important for vaccination. The immune response consists of antibodies. The G, A, and M antibodies occur in different times. You have the CD4 cell and the CD8 cells, and these are what you would try to get with vaccination. If you're infected with SARS-CoV-2, you will get this process open, but it will be about 7 to 10 days after, and many people can die in the process. So therefore, vaccination will try to produce this before the person gets infected, so that the person, even if they get infected, would only get a mild infection, not a severe infection. Age is important. The older the person, the immune response can be more dysregulated and it can be dysfunctional. And this is important. That is why you have to protect the older population early and not delay their protection for long periods of time. And that again is obtained by looking at the pathogens of the condition over the period of time that we have studied it. So that is a quick overview of some of the immune aspects that we have to take into consideration in the infection and in vaccination. The variants have come in big time from autumn of 2020, and we have known quite a lot, and quite a lot has been written about the different variants. At the present, you have variants of concern, variants of interest, and the variants of concern are alpha, beta, gamma, and delta as per the WHO classification. And then you have some variants of interest. Now, what is the effect of the variants? The issue is many changes occur in the virus, close to 5,000, but not all of those are important. However, some of them are clinically important and they can affect one or more of these following processes. That is, they can affect the spread, the transmissibility, they can cause more severe disease, they can affect diagnostic tests, therapeutic agents, or they can affect, importantly, vaccine-induced immunity. And that's something that really interests scientists around the world. Does it affect vaccine-induced immunity? So those are the potential effects, 5 potential effects. So the variants of concern, alpha, beta, gamma, delta, the variants of interest. So the first slide produced by the WHO, but then subsequently other variants of interest have also come in, such as mu and delta. If we look at the delta variant, which is the one that is circulating widely around the world and is affecting so many populations, including Sri Lanka, they were initially called the double mutant because they had 2 critical mutations in the receptor binding domain, the place where the virus binds to the ACE receptor and enters the cell. Not that they had only 2 mutations, but these are 2 critical mutations. That was why they call the double mutant. They have several other mutations in the genes of the virus. And this was the article that just coming out that we described in detail the Delta variant of SARS-CoV-2 and its effect on the vaccination, because you find that the antibodies that are produced from vaccinations are the neutralization capacity is reduced with the Delta variant compared to the other variants such as Alpha. Still, it's able to neutralize it and prevent severe disease. However, we are having an effect on vaccination with the Delta variant, and there are some breakthrough infection, especially mild or asymptomatic infection that are coming through, which I will describe as it goes on. So there are other variants such as the mu and the lambda variants, which we have described, and so many others. And this is a small article on the characteristics of 2 other variants that people are monitoring very carefully with regards to the SARS-CoV-2 virus. So next we go on to the main part of the talk, that is the COVID-19 vaccines. We have spoken about the immune response, the issues with regards to variant, Delta variant, etc., and mu and lambda coming into the equation recently. So when it comes to vaccines, I told you the vaccines are targeted against spike protein on the surface of the cell, and there are several vaccines that have been studied and many that have been authorized or approved so far. This is a list of the vaccines that some of the common vaccines with their properties, and you can see that at the top you have the mRNA vaccines, the Pfizer and the Moderna vaccine, which are the best vaccines around. Efficacy is very high, effectiveness is very high, and then you have the viral vector vaccines, that's AstraZeneca, the J&J and the Sputnik vaccines, and then you have the killed vaccines, which are the Sinopharm and Sinovac. So you find that the effectiveness and efficacy are lowest in the Sinovac and Sinopharm, while it is highest in the mRNA vaccines. Now, interestingly, the person who first described the mRNA process, the vaccine sort of using mRNA in diagnostics and therapeutic, they won a major prize, I think, yesterday. And when they described the process initially, that lady could not get grants for a long period of time, could not get, her job, she had to leave her job, she was demoted, etc., because they thought this process will never work in clinical medicine. And we can see that over a number of years after that, that it has been a major success with regards to SARS-CoV-2, and it will be important when it comes to cancer vaccination, because that is the next goal that the mRNA process will go into. So we have a whole range of vaccinations that are available for us to use in SARS-CoV-2 infection to prevent it, and that should go hand in hand. That's very important with the social distancing measure. Vaccination alone is not sufficient. You have to have the other measures in place that go hand in hand. Now, this slide shows you that there is infection, which can lead to disease. So you can be asymptomatic, you can have mild disease, you can have moderate disease or severe disease. What do vaccinations do? It prevents severe disease. That's the first thing it does. That's important because all vaccines are doing that well. If you can prevent people getting to hospital, ICU, or dying, that is the first thing with regards to vaccination. But we must know that those who are infected or those who have the disease can transmit the disease. And that is important when it comes to the next few slides. Efficacy is if it's in a clinical trial, effectiveness is in the real world. And we have both efficacy trials published in the major journals and the effectiveness trials coming from the UK, US, Germany, Europe, plus Israel, showing that the vaccines that have been are both efficacious and they are effective in controlling the infection. So very early in December, the UK government looked at this process and they categorized 9 different groups. Why I put this slide is because they went very systematical. The older people, the healthcare workers, people with immune deficiency, cancers, etc., vaccinated very early in the process. By February, they had received both vaccines. And they followed this process and vaccinated a big number of the population, over 40 odd million, have been double vaccinated at the present time. It made an effect because you can see the curves. In November, December, you had the alpha variant circulating very widely, and then with vaccination coming on board, you found that the curve turning and the numbers reducing. We know that Delta is coming now and increase the numbers, but the severity of the infection is low because of lots of people being double vaccinated. You have to get 2 vaccines. 1 vaccine is not sufficient. 1 dose of either Pfizer or AstraZeneca vaccine would only give about 30% protection, which is lower than 50% prescribed by the WHO. And 2 doses will increase the level of protection, both the alpha variant and the Delta variant. So 2 doses of vaccination are essential if you want to get appropriate protection. That is important to remember. Now, the issue is that with the Delta variant coming into the focus, that we know that it protects against severe infection, etc., but there are some breakthrough infections. And some people who are double vaccinated are getting breakthrough infection, but there's a mild infection or asymptomatic. Now, the issue with that is that when they get a mild or asymptomatic infection, the viral load is still high. So that those people can transmit the infection to those who are not vaccinated. This was clearly shown in the Oxford ONS study, and that is important to remember with regards to the non-vaccinated group, because they are at very much higher risk from the people who get breakthrough infections, that's vaccinated people who get breakthrough infections. Extremely important point. So in the Los Angeles study, it was found that unvaccinated people had a 5 times rate, the rate of COVID infection 5 times higher, and 29 times more likely to be hospitalized. So the group that has been targeted now is the non-vaccinated, but that is the group that is ending up in ICU, hospitalization, and death, as opposed to the vaccinated group, population. Now, the other aspect to be considered is, what about the immune response following vaccination? Now, the Pfizer company very early said that the immune response could be waning after some period of time. And there was, the risk reduction in infection was 96 2 months after that, 83.7 4 months after. So there was a reduction in there was a 12.5 point reduction in the risk of infection. So it appeared to be that the immune response was waning. And this was again shown beautifully in the Zoe-COVID study, where they've studied people who had been vaccinated, and they found that there was a waning of the response, which I'll show you in the next slide. So looking at a reasonable worst case scenario, they found that the protection could fall below 50% in some elderly and healthcare workers by the winter. And that is why the UK government at the moment is most likely going for booster vaccination. This is a very contentious issue at the moment, giving everyone a booster 3rd dose, but it will be discussed, and it appears that next week the process will start for the general population, but that is again an issue that has been discussed. So these are the figures that were shown from the Zoe-COVID study. 1 month after the Pfizer, 88%, and then 5 to 6 months, it dropped to 74%. And Oxford AstraZeneca started at a lower dose, a lower level, and then also dropped after a few months. So we wrote an article outlining all these aspects when the 3rd dose was recommended for people with immune deficiency. And at present, both the UK, US, and several other countries are giving patients with immune deficiency a 3rd dose. The timing of the timing in the US, it's at least 1 month after the second dose. In the UK, at least 2 months. And generally, people are going for that period or up to 3 months for the immune deficiency, because we know that patients with immune deficiency and cancer may make suboptimal responses that have been clearly shown in many studies that have been published so far. What about children? That's another widely discussed issue. What about children, vaccination children? Initially, the Pfizer vaccine was approved for children, for persons over the age of 16. And the other vaccines, such as Oxford, AstraZeneca, and the Moderna, etc., were 18 years. Then subsequently, studies came up, and now the Pfizer and Moderna have been approved. The Pfizer has been approved up 12 and over. And it has been rolled out in the US and in Canada and several other countries like Israel. In the UK, the 16-year-olds and 17-year-olds, all 16-year-olds and 17-year-olds are offered the Pfizer vaccine at present. 12 to 15, the high-risk children are given the Pfizer vaccine, but the other children it's still not approved for the other children. So the JCVI, the Joint Committee of Vaccination Immunization, said they cannot recommend for other children. However, the government may override that. And there's again a lot of discussion. In the next week, we would know what is happening. We have to remember, put it into context. In this article, we have described the different aspects of COVID-19 in children, the pros and the cons with regards to vaccination in children and the issues with transmitting to adults. And since children get only mild infection, should we be vaccinating them, or should we get these vaccines out to places where even healthcare workers do not have any vaccination? So these are the issues that come up, and that has been described in these articles, in the article that has been written here. Again, this has been discussed in the sort of current updates, which was again written very recently about vaccinating children. And at the present time, 16 and 17-year-olds are vaccinated in the UK, and the other group, only the high risk, from 12 onwards, only the high risk. Below 12, studies are ongoing, and many studies should be reporting in the next few weeks or in the coming months. So during this quick talk, what I have told you is a quick overview of the immune response to COVID-19 infection and following vaccination, the antibody response, the cellular response, and I told you there is a response that occurs following infection, but it takes some time. That is why we try to give a vaccine to mimic that response so that the person is protected. And then I told you about the variants, including the Delta variant, which is causing some concern about vaccination, although at the present time it is protecting against severe infection, but that the hope is that no other more nasty variants arise in population, that the transmission is very high. And that is what we should focus on, reducing transmission in areas that are not protected at the moment. And then I spoke to you about the COVID-19 vaccines, the ones that were approved, the mRNA vaccines, the viral vector vaccines, and the kill vaccines. And I told you some of the properties of those, vaccinating patients who are immune deficient or who have cancer, vaccinating children, and then giving this third booster in a wider range of people, which is again being discussed, especially with the waning of some immune responses that are seen. And when it comes to children, we have to balance the benefit to them as opposed to benefit to the community. Thank you very much.
Thank you, Professor Ranjit, for your elaborate and up-to-date lecture. And we'll be again getting on to the questions at the end of all three speakers' talks. And virtual participants are reminded that please type your questions in the Q&A box, and we will direct them to the relevant speakers at the end of the symposium. So thank you, Suranjit, and we'll get on with the next talk of this symposium. It is named Simply a Piece of Bad News Wrapped Up in a Protein, Sri Lankan Perspectives. to be delivered by Dr. Chandima Jeevandara. He's the Director of Allergy, Immunology and Cell Biology Unit in the University of Sri Jayadulpura. Over to you, Chandima.
Thank you, Chairperson, sir, Madam, for the kind introduction. It's indeed a great pleasure to be with you to talk about the variants of SARS-CoV-2 and the disease trends, and also our work from the Allergy, Immunology and Cell Biology Unit of the University of Sri Jayawardhanapura, Sri Lanka. And I'm indeed grateful for the President and the Council members of the Gold Medical Association for inviting me to talk to this prestigious audience. A virus is simply a piece of bad news wrapped up in protein. The biologist Jean and Peter Mendever wrote in 1977. In January, scientists in China deciphered the most important news in 2020. They unraveled the genome sequence of an unknown pneumonia in China. No sooner Sri Lanka reported its first case of coronavirus back in January 2020 from a foreign national. The first local coronavirus case was detected on 11th of March from a laboratory. The dawn of the year 2020 transformed our lives, and more than 221 million confirmed cases of COVID-19 and more than 4.5 million deaths reported to date. And this is a gross underestimate. It has crippled economies, disrupted lives, livelihoods and social lives, and increased inequality was evident, especially in terms of vaccination. Let's talk about the situation in Sri Lanka. Like the other parts of the world, Sri Lanka is not doing very well at the moment. We report quite a significant number of cases, and there's a slight decline over the past 1 week. The most important finding here is the number of deaths. The 7-day average of the number of deaths is staggeringly high and probably one of the highest in the world, which is not very good news as one of the best countries in terms of healthcare. However, there are positive sides. Our vaccine rollout is extremely good. We are even catching up Americas right now. And if you can see, even in the global map, Sri Lanka is becoming dark green, which is great news. And we are hopeful that we will be able to contain the case numbers at least by mid-October. So, ladies and gentlemen, in that background, let me take you through our Sri Lankan experience in genomic sequencing and the users and the values of sequencing globally for future control. SARS-CoV-2 virus is a single-stranded positive-sequence RNA virus which belongs to lineage B of the genus beta coronavirus family. It shares about 78% homology with the SARS-CoV virus, and it consists of 2 open reading frames, namely ORF1A and ORF1B, which constitute for about 2/3 of the genome. The rest 1/3 of the genome encodes for 4 structural proteins, namely nucleocapsid, spike, membrane, and envelope. So altogether, there are about 30,000 base pairs, which we unravel through genome sequencing. So back in 2020 March, Sri Lanka did not have sequencing facilities. However, Allergy, Immunology and Cell Biology Unit of University of Jagannathpura had the facilities, had the expertise. So Minister of Health invited us to be part of this effort in identifying this SARS-CoV-2 genome, and we were very happy to contribute onto their request. And we stepped in. Genome sequencing, we know it originated in late 1970, when Frederick Sanger at the MRC Center in Cambridge developed a gel-based method that combined DNA polymerases with a mixture of standard and chain-terminating nucleotides known as DNTPs. Since then, this technology has advanced rapidly. As of today, even in our lab, we have both second generation and third generation sequencing platform, which is capable of sequencing the SARS-CoV-2 genome. There is a notable difference between the two platforms. Let's look at it briefly. The second generation platform is able to generate short reads, but through this method, when we develop the draft genome, there may be sequence gaps, and missing sequence leads sometimes limits the biological interpretation. To combat the practical issue, Oxford Nanotechnology developed high-five reads, which are using in our laboratory currently, and it is capable of encoding the complete genome. A comprehensive structural, functional, and organizational picture of the genome is possible through this method. Let's compare the speed and the scale between genomic responses in SARS epidemic versus SARS-CoV-2. Back in 2002-2003, only 3 viral genomes were publicly shared within the first month, and only about 31 were available within 3 months. However, things are really different in 2021. The SARS-CoV-2 pandemic, during the SARS-CoV-2 pandemic, the causative pathogen causing this unexplained pneumonia was revealed within 1 week. And the pathogen was announced as novel coronavirus and then later renamed as SARS-CoV-2 in the beginning of January 2020. 6 genomes are publicly shared within a couple of weeks, allowing rapid development of diagnostic assays and strategies for many public health interventions. And as of today, more than 2 million SARS-CoV-2 genomes has been uploaded to the world databases. It's mainly because of the third generation sequencing platform, which even possible, even in a failed setting. This is a picture, I took it from the net, where their sequencing is happening in a failed setting. This is in our lab. Our scientists are working using a similar platform, which helped the sequencing is accessible to even small laboratories with providing real-time analysis. So how we have responded? So we sequenced our first genome on 23rd of April 2020 using the second generation platform, which was available with us at that time. So the first genome was introduced around this day, and then until the second wave, which we observed from associated with the garment factory, we did not do much sequencing because there were no cases. We have updated only about 16 genome sequences until about October. But since then, we detected significant number of cases in the country, and we increased our capacity to about 94 samples per month. And then later, during this epidemic, this phase of the pandemic, we increased our sampling, and we are now currently doing about 94 samples a week, probably the maximum. Let's go back and evaluate our performance compared to the other parts of the world. At the beginning, we were a bit late to upload our sequence, simply not because of anything, because At that time, back in 2020, we did not have necessary reagents and consumables in Sri Lanka. To get it down to Sri Lanka, it took some time. But since then, we have sequenced more than 1,200 genomes. Compared to the developed world, our performance is grossly inadequate. However, when we compared our sequencing capacity in the region, we are doing a very good job and probably the best sequencing facility in the South Asia. And even in Asian region, we are around number 6, which is very good news from a single sequencing facility. Operational structure, unfortunately, we don't have an operational structure as such. So if you compare with the UCOG UK, where all sequencing platforms are combined, it's working as a unit. But here, whenever we want to do a sequencing run, we need to contact in each and individual labs or the regional laboratories to get down samples to get a clear picture about Sri Lanka. The SARS-CoV-2 virus evolves over time. So from one generation to another, similar copies are generated. It's like an old photocopier. Sometimes from the parent generation, when it develops children, it will not produce the exact similar copy. There are mutations. And certain mutations may provide an added advantage to the virus, like increase in transmissibility. So that's why WHO has named 4 variants of concerns. So when you talk about a variance of concern, it could either provides advantage to the virus in terms of increasing transmissibility. Maybe it will cause severe illness, and probably it affects the vaccine efficacy, and sometimes it might impair the naturally acquired immunity and also might interfere with the diagnostic assays. So based on that, and after carefully evaluating the spread of different variants, they provide, WHO gives different levels of attention to these viruses. So that's the most cautious one is the variants of concern, where we have, as said today, we have 4 variants of concern, namely B117, this is the UK variant, B1351, this is the beta variant found in South Africa, this is the gamma variant found in Brazil, and this is Delta, first detected in India. And since its first detection, it has branched out and it has developed many variants of concerns and 5 variants of interest. The latest addition is the mu. So all governing bodies are closely monitoring the phenotype of these variants. So when the virus evolve over time, there were hostility corpus. As you can see here, at the beginning, there were only one mutation provided an advantage to the virus, which is the D614G mutation, which is supposed to be facilitating the virus's increased transmissibility. Sooner, everywhere in the world, virus acquired this mutation, becoming the dominant variant causing SARS-CoV-2 virus viral infection in the world. But later, after some time, we saw certain key spike mutations happened, giving an added advantage to the virus. For example, in alpha, it clearly showed that it is more transmissible, at least 50% more transmissible than the Wuhan variant. And in delta, it is 50% more transmissible than the alpha, and it is 100% more transmissible than the Wuhan variant. And also, compared between alpha and delta, alpha is still considered to produce more severe disease compared to delta, although the transmissibility is far higher than alpha in delta variant. So in Sri Lanka, our sequencing has become a critical tool, not only in detecting and characterizing viral pathogens, but also supporting infection control and also making key epidemiological decisions. Let me do some examples. When we detected the UK variant in Sri Lanka, there were restrictions in international travel for travelers. Then when we found out that the UK variant is the causative SARS-CoV-2 lineage for the ultra fast spread in April, we went into a lockdown. And then we found that Delta detected in Sri Lanka, the health ministry supported a decision of banning interprovincial travel because we detected them mainly in Colombo. And then later we found out that Delta is spreading fast all over the country, which again led to a decision of closing down the country. So these are important findings to make policy decisions in Sri Lanka. Although the timing of the decision was not the most appropriate, we can be happy that we have done our part. We have uploaded our data to the World Database, and as of today, GSAID is a recognized sequencing database where we upload our sequencing data to the World Database. So it has quite a significant number of Sri Lankan SARS-CoV-2 genomes uploaded. If we talk about the emergence and spread of Delta in the country, at the beginning, back in March, April 2020, most of these cases were imported from different parts of the world. Then we had our own Sri Lankan variant called B1411, which was replaced by Alpha and then Delta sequentially. Then we went on to check the spread of Delta in the last week, and we found that all provinces had a predominance of Delta as shown in this blue color. Not only that, we found several key mutations which is not seen elsewhere in the world. These are the 3 key mutations in this protein which I am listed here. Apart from that, there is another mutation also which we think will be important in the future, also found in the non-structural region of the virus. Then we further evaluated the emergence and spread of this mutated Delta as shown in this yellow color. As you can see here, from the parent Delta, this is the original Delta and this is the mutated Delta. It branched out as a different lineage, and we wanted to find out whether is there any transmission advantage with the mutated Delta, and at the initial runs it looked like, but then it stabilized, and from our latest run we don't seem to observe any higher transmissibility compared to the original Delta we saw and we see from other parts of the world. However, there is one key mutation which everyone is interested in, that is A701S. In the S gene, and out of the worldwide prevalence of 1,082 mutations in this particular region, Sri Lanka has reported 30%, which is the highest contribution from any country in the world. And even Even the last 60 days, Sri Lanka has contributed more than 75% of the ST mutations in the world, which is not very good news. As you can see here, compared to the other country, Sri Lanka stands tall here in this map with regard to ST mutation over the last 60 days. And in our unit, as I mentioned earlier, we had our Sri Lankan variant, then we had the UK variant, and this is Delta. And you can see the disease trend and the deaths associated with different variants, the Sri Lankan variant, the UK variant, and the Delta variant. Furthermore, there are recent studies to show that the vaccine-induced immunity can be escaped, especially with Delta variant. And these are important findings to decide on certain policy decisions on vaccination. So, ladies and gentlemen, WHO has, in a recent document, identified 5 key applications of genomics sequencing in SARS-CoV-2. Even though we have not contributed to the first two, that is the understand the emergence of SARS-CoV-2 and understand the biology of SARS-CoV-2, we have contributed. We will probably be contributing, improving the diagnostic and therapeutics by way of our genome sequencing. And most importantly, for Sri Lanka, we have been contributing significantly to the investigating the virus transmission and spread, and also to decide on certain policy decisions for epidemiology unit. Not only that, we have published, and this publication will appear very soon as an open access article. So, ladies and gentlemen, we need to learn from past, and we need to predict the future. Although the world is moving towards vaccination and about to reach herd immunity in certain parts of the world, I don't think we will have an end to this pandemic. The new variants will emerge, and we don't know how bad the next emerging variant will be. So we need to prepare ourselves. And in view of that, WHO has produced this policy document back in January 2008. And I'm happy to note that MRI and University of Colombo, they combined together in the effort of genome sequencing. And I think they have issued their first report of genome sequencing in the past couple of weeks. So as I mentioned earlier, as the goal of reaching herd immunity, vaccinating so many people that the virus simply has nowhere to go with the emergence of Delta, I realize it's just impossible to reach that. And this is our sequencing team. We are very grateful, and our team and the research funding organizations. Thank you very much. I'm happy to take any questions if you have.
Thank you, Dr. Chandima Divandara, for the excellent lecture on Sri Lankan sequencing. And also, I would like to congratulate your team for the excellent work. So we'll leave the questions to the end of the session. And let me introduce the next speaker, Dr. Ananda Vijayakrama, consultant physician, the National Institute of Infectious Diseases, Colombo. He will be talking on COVID-19, treating severe cases. Over to you, Dr. Ananda Vijayakrama.
Good afternoon, ladies and gentlemen. First, I would like to thank the President and the Council of the Gold Medical Association for inviting me for this academic sessions and giving me this opportunity to share some of my experience on this important topic. About a year ago, I came across this article published in a Nature journal, which gives a prediction of the future of the pandemic. It started by saying June 2021. This was written in August 2020. The world has been in pandemic mode for a year and a half. The virus continues to spread at a slow burn. Intermittent lockdowns are the new normal. The approved vaccine offers 6 months of protection, but international deal making has slowed its distribution. An estimated 250 million people have been infected worldwide, and 1.7 million are dead. When I read this, I just laughed at this. I thought it's not believable. It's too much of imagination. However, when we looked at the historical accounts of initial estimates of deaths, there had been a quite a high, relatively high mortality rate associated with this infection. In China, estimates showed about 3 to 5% of mortality rate, changing across their regions. And a WHO study provided a mortality rate of 3%. And in hospital admitted patients, the mortality rate was much higher, which was shown in clinical studies too. In solidarity study, the death rate was around 12%. In Rendezvous study too, the death rate was around 7 to 12%. In the recovery study, it was much higher, around 24%. We had quite a large number of deaths reported across the world. However, we had a lot of patients. We had very few deaths. So we thought probably we are doing well or better than other people. This 94-year-old gentleman was transferred to us from a private hospital with a history of progressive shortness of breath, fever, and cough. And he was hypoxic on admission, but improved with oxygen. And he was hemodynamically stable. Later, he became confused. His saturation still maintained with 2 liters of oxygen. His creatinine was elevated. Other parameters are generally okay. The chest X-ray showed patchy consolidation in all 3 zones of right lung and mid zone, suggestive of COVID pneumonia. This is his first chest X-ray. It showed the involvement of right lung mainly, and the second X-ray showed the involvement of the left lung too. And because of his confused state, we did a CT scan of the brain too, which showed age-related cerebral atrophy. And the HRCT showed again changes suggestive of COVID pneumonia, but in addition, it showed changes suggestive of secondaries of neoplastic lesion, with the bilateral pleural effusion, small pleural effusion, and bone destruction with the nodular opacity. However, in spite of all this 94-year-old gentleman gradually improved, recovered, and went home after 2 weeks. So this prediction came from this Nature journal. I thought probably not acceptable. Their imaginations were too wild. But then what we saw at the time they mentioned June 2021 was beyond their wildest imagination. We had 225,000 cases as of now, more than that, and more than 4.6 million deaths is by 13th of September. And these 2 graphs shows the occurrence of daily new cases in the world and the deaths. You can see the high numbers occurring in waves. And when it comes to Sri Lanka, these are the deaths reported by the Ministry of Health from 1st of April. And we see the number of deaths which were quite low at the beginning of April this year went up to more than 200. And we had large number of deaths during the last 2 months. So then with this, we face the challenge with what to treat these patients who are critically ill, severe cases. Again, if we go back to our old gentleman, how did he recover? Did we give him monoclonal antibodies? Did we give high dose of dexamethasone, tocilizumab, remdesivir, ivermectin? No, we didn't give anything. He just had standard treatment with oxygen, self-proning. He had secondary evidence of secondary infection, so we treated him with antibiotics, enoxaparin, and his sugar was high, so we gave him insulin. And that's all. And he recovered. If he was given anything like high dose steroids, very high dose steroids, and or monoclonal antibodies or anything, then his recovery, which probably would be little unlikely with all this age and other complications, would have attributed to that particular drug. And that is why we need evidence. That is why we need trials. Interestingly, a large number of trials have been started. Even by early March, more than 300 trials have been registered on medicines for COVID. And you can see worldwide, more than 4,000 trials had been registered by end of December last year. And majority of these trials are in Europe and North America. You can see in spite of the initial outbreak occurring in China, and in spite of having all these traditional medicines in China and in India and in Sri Lanka, the number of trials conducted in the Asian region is quite low. So many agents have been tested: antivirals, anti-malarial, antibiotics, antiparasitics, monoclonal antibodies. However, they have other issues in spite of all these studies. Many preprints of studies which are not peer-reviewed are available in the web. Data in some of these preprints have been used for meta-analysis, and then some preprints were later withdrawn. And some drugs used in clinical trials were promoted to use. This is the infamous article on hydroxychloroquine, which was retracted, which was published in the Lancet surprisingly, and then at the same time, another article published in JAMA was withdrawn. This is a science article in the Science Journal. Two elite medical journals retracts coronavirus papers over data integrity questions, which is very probably maybe the first time in the history of these journals such withdrawal occurred. And then this preprint of ivermectin, which again was withdrawn. And it is important to mention that this article was considered in a meta-analysis. And then we see a cocktail of drugs being used in our country: vitamin C, vitamin D, zinc, azithromycin, doxycycline, dexamethasone, ivermectin. Many of these drugs had been used without much evidence. However, we have this therapeutic guidelines by the WHO. In the group, I also was fortunate to be there. And the current recommendations in a nutshell are these. The recommendations against systemic corticosteroids in patients with non-severe COVID-19, a strong recommendation for systemic corticosteroids in patients with severe and critical infections, so that is hypoxic patients. So this guideline specifically mentioned not to use corticosteroids in non-hypoxic patients. Conditional recommendations against remdesivir, against lopinavir, against HCQ, against ivermectin, and strong recommendations use IL-6 receptor blockers, tocilizumab or serlulimab. If you go to a case history, this 34-year-old male weighing 130 kilos was admitted to our hospital just a few weeks ago with fever for 8 days duration. When he was admitted, his saturation, he was brought in an ambulance with oxygen or 5 liters of oxygen. Still, his saturation was 85%. We increased his oxygen with NRBM later, which we had to increase to 15 liters. Started on dexamethasone 6 milligram IV, enoxaparin. He was getting desaturated even with mild exertion, so we had to catheterize, advocated complete bed rest. These are his X-rays. The left-hand side one is the on admission, and the right-hand side one is 4 days later. You can see how the lung involvement has progressed. And this his monitoring chart. The blue line shows the oxygen supply. This the oxygen 15 liters. And the red one shows the oxygen saturation. Oxygen initially his saturation was around 83%, which improved to 95% and 97%, but then it dropped rapidly in spite of having 15 liters of oxygen. So with that, we gave him tocilizumab. Also, when the oxygen saturation started dropping in spite of having giving 15 liters, the response was not enough, not poor, and we had to intubate and ventilate him on 20th, the third day of admission. This is chest X-ray showing extensive involvement of both lungs. And then, in spite of all this, he gradually improved. And we were able to extubate him on 27th. He was still oxygen dependent. We transferred him to a medical ward on 31st of August. Urine culture became positive for Candida. He had persistent tachycardia. His echocardiogram was normal, and he did a CTPA because of this persistent tachycardia. And these are the CTPA showed. A filling defect on the right pulmonary artery, and then there are some more filling defects in the branches. So he has several segmental, several pulmonary emboli affecting segments of the right lung. Now he's on enoxaparin and we have started him on warfarin 2. We are adjusting the dose of warfarin. He's without oxygen now. We are mobilizing him slowly. He has lost 30 kilos during this period. Now he's 100 kilos of weight, but he's gradually improving. At this juncture, I thought it is worth mentioning something about tocilizumab. This text message I received yesterday. If anyone is looking for tocilizumab, within brackets, last moment injection to recover life, below is the contact received from a friend in pharma. So the details of the contact was given. Unfortunately, an idea had been given to people and even to some doctors that it is a magic bullet. It's not so. Of course, it is very useful in some selected cases. But on the other hand, it can make the disease process worse if not used correctly. This is the WHO living guideline recommendation. Either 6 receptor blockers reduce mortality and need for mechanical ventilation based on high certainty of evidence. Low certainty evidence suggests they may also reduce duration of mechanical ventilation and hospitalization. The in-hospital mortality is reduced in the REMAP study. However, if it is given to 100 patients from whom it is indicated, 8 to 9 patients who would otherwise die would be saved. But you have to realize that still 27 patients in this group still would die. You cannot save everybody with tocilizumab. However, there's a significant reduction of death rate when it is used correctly. This is a temperature chart of a patient who later died. I was listening to the presentation of the death review by a SHO of that unit. The SHO started saying, when he was presenting, said the patient was given tocilizumab with broad spectrum antibiotic coverage. So obviously patient had high fever. His her procalcitonin was high. In spite of that, patient had been given tocilizumab, maybe because of this idea that it is a magic bullet, and patient went into severe sepsis and died. So it is important to use this important good drug appropriately. Unfortunately, we have been seeing an abuse of this drug due to improper or propaganda given by various people. This 45-year-old diabetic, again admitted a few weeks ago, was hypoxic. He had been on oxygen at home. He is a diabetic with peripheral vascular disease. He had below amputation of one side. And we had to increase his oxygenation to non-rebreathing mask. Then we were alternating his oxygen supply with high flow nasal oxygen and CPAP. We had to ventilate him on 24. This is how his X-rays progressed. The left-hand one is the on admission X-ray. And then in the ICU, we had many issues. He developed infections, secondary infections, hyperglycemia, hypernatremia. His sodium increased up to 168. We treated him with desopacine nasal spray. It didn't work. Then with advice from the endocrinologist, we changed to oral desopacine, and we started him on low dose desopacine. Then he started developing ischemia of the good leg too. In spite of all that, we extubated him because his respiratory wise he was improving. And then we noticed that he had developed left-sided hemiparesis and aphasia. And this is the CT scan done, which showed multiple infarctions in the brain. This is another issue we see may not be common, but then attributed to the severity of these patients. Again, we had to intubate this patient because of a lot of secretions. He went into again respiratory distress. But now again, we are planning to intubate him. But then probably we might have to, he might have to have surgical intervention for this because his part of the leg is getting gangrenous. It is important to mention something about the steroids too here. On the basis, how it is recommended on the basis of preliminary report from the recovery trial, the COVID-19 treatment guideline panel recommends using dexamethasone 6 mg per day for up to 10 days or until hospital discharge, whichever comes first, for the treatment of COVID-19 in hospitalized patients who are mechanically ventilated and in hospitalization patients who require supplemental oxygen but who are not mechanically ventilated. So in hypoxia patients, the indicator is useful. And the panel recommends against using dexamethasone for the treatment of COVID-19 in patients who do not require supplemental oxygen. We see abuse of these steroids. The high death rate we saw in India was attributed by some people to abuse of these steroids. I don't know how much it is contributing in Sri Lanka too for deaths. We see the prescribing of dexamethasone to patients with mild illness, prescribing of very high doses of dexamethasone for patients with severe disease, continuation for steroid for a longer period, longer than recommended. High dose steroids, we know, result in poor blood sugar control, which affects the prognosis of the patient. Patients get hypokalemia, which is a common thing we see. The secondary bacterial infections, second fungal infections. So this steroids again has to be used judiciously. Then we have to talk about Rendezvous. If I fail in my duty if I don't talk about this. The NIH guideline recommends use of Remdesivir and dexamethasone in patients who require supplementary oxygen. However, the WHO therapeutic guideline does not recommend use of Remdesivir. In fact, it recommends against using Remdesivir because of lack of evidence. If you go through the evidence for Remdesivir, which I'm not going to highlight here, you will realize that the evidence are very, very minimal. It is not cost effective using Remdesivir, in my opinion. Another drug which was promoted, though not for severe cases, is the antibody cocktail of tezepelumab and imdesivimab, which is popularly known as Regenco, which was given to apparently given to Trump. Now the studies show that it is effective if it is given to 100 patients. Sorry, there's a missing letter. 3 medical visits can be prevented if it is given to 100 patients. The one dose is around 100,000. So for 100 patients, we will need 100,000 into 100, and we can prevent 3 medical visits. So how cost effective that has to be considered. This is for patients who are non-severe. So we face issues like the lung inflammation, thrombosis of pulmonary vasculature, thrombosis in other vessels, secondary infections, bacterial, fungal, and then bleeding. And some of these could be iatrogenic also, bleeding, so hyperglycemia, worsening or new secondary infections, electrolyte imbalance, and some of these could be due to drugs or treatment we give, especially steroids. But we have some weapons to fight this. Yeah, oxygen probably that's the best weapon, I believe, to fight this. Yeah, we can use give it using nasal high flow, use prone ventilation, and then of course we have to use steroids correctly, anticoagulation, IL-6 inhibitors again correctly, antibiotic, monoclonal antibodies, remdesivir, ivermectin. Those are I put questions. I have already mentioned about this. So out of these, I believe it is very important to detect hypoxia early. If we treat these patients early with oxygen, correctly with oxygen, whenever necessary using non-invasive ventilation, we can reduce the mortality and morbidity too. So for this, it is important to have a place where we can observe people. I don't believe in using CCTVs. I don't think we can use CCTVs in monitoring these patients. The parameters, especially the respiratory rate, we have in our hospital wards like this, open wards like this. This, of course, we were talking to some of these patients. So we can easily observe these patients, monitor these patients. When we go in, of course, we wear full PPE, but outside the wall, we just wear a face mask because we know that it won't spread otherwise. So then I think it is important to have timely tracheostomy. Again, getting a tracheostomy can be difficult. So in our hospital, we started using a percutaneous tracheostomy. This is on the right, on the left corner is me who is performing the percutaneous tracheostomy, and one of my senior MOs is using a amboscope to guide the tracheostomy. And then we have other things like sedation holds, trying to attempt to early weaning so that we can get them out of the ventilator quickly. And then again, it is important to do frequent cultures to detect bacterial organisms or fungal organisms which infects patients secondarily, which is again a common problem. And of course, the psychological and nutritional support for these patients is important. In spite of this, we have problem. This 57-year-old doctor was transferred from a base hospital on 16th of August with hypoxia. He had symptoms for 15 days, that positive on 3rd of August. Actually, initially he was admitted to our hospital, but he left on his own on the same day. He said he will come back if he has problems. And when he came, he was hypoxic on 15 liters, saturation was 89%. So we put him on NRBM, and then he gradually improved. His procalcitonin was high, platelet was towards the low side, D-dimer was little high, and chest X-ray showed the progressive stage of COVID pneumonia. And the HRCT showed moderate degree of COVID pneumonia. He had no pulmonary embolism. And we continued him with enoxaparin, dexamethasone, antibiotics. We gave high nasal oxygen using NRBM, then high flow, then deep from time to time. And he improved gradually. Then we were able to take him to 60% of the injury mass, and then we transferred the patient to a medical ward from the ICU. 2 days later, he was complaining of severe pain down his legs. In fact, he said the pain is so bad, he feels like committing suicide. We immediately got our radiologist who came and did using the portable scan, did a Doppler scan, which he thought is suspicious of DVT. He was anyway on enoxaparin. However, we planned to get a CT scan done next day. Next day morning, we found his urine output was reducing, and we did a CT scan, which did not show any deep vein thrombosis. Instead, the CT scan showed bleeding. He had bleeding into psoas muscle. You can see the size of bleeding, and this is another view. It shows multiple sites of bleeding into psoas muscle and the retroperitoneal muscle. He was getting dyspneic. We took him to the ICU again, and within half an hour, he collapsed. We transfused him with several pints of blood, but unfortunately, he didn't recover. He died. So in spite of all the weapons, we have all these problems. So we need more knowledge. I believe for this, we need documentation of the success with our usual case management, about unusual cases. Then people may be using off-label drugs, but then we need to document those to see the response to those. Then only we would know whether those will be useful or not. Otherwise, after 6 months of using this off-label using of drugs, still we will have the same knowledge and or lack of knowledge. And it is important to share experience about what problems we face, what solution we use. Then only I think we would be able to overcome the problems faced in managing severe cases of COVID. Thank you very much.
Thank you, Dr. Vijay Krama, for your excellent clinical lecture. I think it will be very useful for clinicians managing COVID patients right around the country. Now we have, thank you very much again for all three speakers. There's 15 minutes left. That was we are on schedule. So we have 15 minutes left for Q&A session. And we have received a couple of questions. Let me first direct, this question was asked from the director to Professor Ranjit. Can you elaborate more on the current recommendation of vaccination for children in UK? Can you elaborate more on the current recommendation of vaccination for children in UK? Because in Sri Lanka, we are about to start the vaccination for children. I think we are eagerly waiting what experience the UK clinicians and the community had with vaccination on children.
I suppose that's for me, Tilak. Yeah. So now a very, very hotly contested sort of topic in the last few weeks because of the pros and cons given by different people with regards to vaccination of children. The present position is that 16 and 17-year-olds, all 16 and 17-year-olds would have been offered the vaccine. Then initially they offered it to high-risk children. There were about 4 categories of high-risk 12 to 15-year-old children who were offered the vaccine, and that was to be the Pfizer vaccine. A few days ago, 2 or 3 days ago, the government medical officers, that is the chief medical officers, overrode the decision made by the JCVI and said that the children between the age of 12 and 15 would also be offered Pfizer vaccine. They said one Pfizer vaccine. That second dose would be put off till next year, the option of second dose. And the children, the parents would be asked to give consent in 12 to 15. However, the child can override the parents' decision if there is a discrepancy. With 16 and 17-year-old, the child's perspective would be taken, because at that time there was the rule that at 16-year-old, if they are competent to give the consent. So that is the current position with regards to child vaccination in the UK, and it's a Pfizer vaccine. Moderna has also been approved, but it's a Pfizer and Moderna. We know that the AstraZeneca is not given to anyone under the age of 40 in the UK.
Okay, thank you, Saranjit. I hope it answered the question of the pediatrician who raised the question. And there's another question. I am sure this is also from Professor Ranjit. Would you recommend a different brand of a vaccine as a booster? I want to add that to that. What is the basis for that? Would you recommend a different brand of a vaccine as a booster? That again is quite relevant to Sri Lanka, because at least for the healthcare workers and the high-risk group, there's a talk that third or the booster is on the way. So would you recommend a different brand? Yes. And what is the basis for that?
It depends on, so in the UK, again, the JCVI just put their recommendation out that all over the age of 50 would be offered a third booster dose. About a week or 2 weeks ago, they said that people with immune deficiency, significant immune deficiency, would be given a third dose, and that was to be the Pfizer vaccine. A few days ago, it was all people over the age of 50 to be given the Pfizer vaccine. And the reason, even if they have received 2 AstraZeneca vaccines, or if they have received 2 Pfizer vaccines, the third dose was to be a Pfizer vaccine. And even for the next year, 2022, they have ordered 30 million doses of the Pfizer vaccine to be given, if needed, if that need originates to people as a fourth dose or whatever. So now, why is that? Because there is, we all know that the mRNA vaccines are the best vaccines around. We do know it. And the viral vector vaccines come lower than that, at least at the original studies. And we know that when they did the mix and match studies, we found that people who received 2 AstraZeneca vaccine, the antibody response was lower than those who received the AstraZeneca vaccine followed by a Pfizer vaccine. There were a bit more adverse effects and people had to miss 1 day or 2 days of work. However, the antibody response, at least the antibody response, was definitely better. And that was again confirmed by a publication on this Thursday in the New England Journal, where the group from Sweden again did that, because the problem was that under 60 or 65 in Europe, you would not get the AstraZeneca vaccine. They would not give it. In the UK, it was only 40. And in that group, some healthcare workers had also already received the AstraZeneca vaccine. So there was a group of people who received the Pfizer, and it was up to 115 times higher, about more than 100 times higher immune response, antibody response at least, in people who received the AstraZeneca followed by the Pfizer vaccine, as opposed to AstraZeneca. Now that is a 2-dose vaccine, but the 3rd dose vaccine, the UK government at least, and the JCVI has taken the option of going with the Pfizer vaccine. However, I must preface this because that is very important. It all depends on what resources you have at the time, because if you can't get Pfizer, and if Pfizer has to be given to the cancer patients, etc., then you have to individually, country-wise, decide which vaccine, because we know that for the booster, both AstraZeneca and Pfizer has been approved. So that has to be taken into consideration, because it is not only one consideration about what vaccine you want to use, you have to mix it together with what is available. Because when a booster is needed, it's far better to give the booster with what has been approved rather than waiting for one specific vaccine. And that is an important point that should also come into the equation.
Right. Thank you, Suranjit. I think I have another question from Dr. Jivandar, because the final comment you made is that about the Delta variant and its increased transmissibility. Now, as a clinician or community health point of view, this is my personal view. I just want your opinion on that. Now, when the virus is highly transmissible, Delta variant, and it is relatively less clinical severity, will that boomerang on its very existence in future? Because now we see many more people are getting infected, and even though the high-risk people are getting complications, but among the children and the young, healthy people, they become seroconverted even without the vaccine. So will that be a detriment for the existence of the virus in future? So the mutation became sort of a boomerang on the virus itself. Is that a hypothesis or it's a plausible?
Well, that's an important point. For example, when the virus spreads within a given community, it increases the chance of mutation, like we saw, especially in the Colombo Municipal Council area, where we saw 3 spike mutations appearing in our Delta variant, and which was alarmed by several international bodies to us also. And probably with these, especially with the high vaccination drives and high infection rates, especially it may spread among the unvaccinated people. Because of the high transmissibility, because of the high, the virus can replicate because of the high transmissibility, there are chances of more adverse mutations appearing, especially in a setting like ours. And on the other hand, as you said, there may be quite a lot of asymptomatic infections among children, which might produce immunity towards COVID-19, especially with Delta variant. So those are the questions unanswered. Probably the future of COVID would be something like that. We will have a situation with mutations, and which will have some stability to the virus, and probably will not kill people as we saw in the past. So a stable virus will remain with us for a longer time.
Right. Thank you, Doctor.
Doctor, there's another question to Dr. Jeevandara. Will there be late complications of vaccine? I think there are quite a few videos going viral in social media on some adverse effects. That might have been the probable root cause for this question. I would like to get your opinion, please.
Well, the COVID-19 vaccination drive is only maximum 1.5 years old. And we have already observed the side effects after administering millions of doses. And there are certain side effects, so very rare side effects associated with immediate side effects. But long-term side effects and the side effect profile probably will, even after 1.5 years, we have not heard of major side effects. So I don't think there will be major side effects on the long run also. So it's again, we cannot definitely say there will not be any side effect, but there is, everyone is monitoring, especially the post-marketing surveillance are on to understand or detect any any side effects, if any. But in my opinion, it's very unlikely that we'll have a major side effect associated with the vaccines.
Thank you for the reassurance answer. So next question is to Dr. Ananda Vijayvikrama. Now, there is a concern that AstraZeneca is prone to, there is a tendency to cause clotting. So now presume that you are going for a third dose of a person who's had 2 Sinopharm vaccine with the AstraZeneca. There is a question, and they just asked what your opinion on this as well.
Well, actually, I don't know. Because I don't think any country has that experience after 2 Sinopharms giving another AstraZeneca dose, whether that can increase clotting. I don't know whether Suranjit has any details from some other country. What we saw was there was increased incidence of clotting reported in patients, especially with the first dose of AstraZeneca, but that was not shown with the second dose as such. That was especially in the younger population. But if it is given as a booster dose, I don't know whether it can cause the same thing or worse thing or less clotting.
Thank you. I think we haven't taken the decisions as yet on the third dose and which vaccine we are going, if we are going for that. So let me, I think I would like to, since there is absence of further questions, I would like to conclude the session, and I would like to thank all 3 speakers for elaborating on all the important aspects of COVID infection management, prevention, and all the aspects of diagnostics as well. So in addition, I would also like to thank the virtual audience who joined and for their active participation. So on behalf of Gall Medical Association, I would like to thank all the speakers and the audience. Thank you very much.
Thanks.
Thank you, co-chairpersons. Next in line, we have the 4th symposium, Evolution of Dermatology, to touch or not to touch. I take the pleasure of inviting the co-chairpersons of the session, Dr. R.W. Kodikararachchi, Consultant Physician, Teaching Hospital, Karapitiya, and Dr. Binari Vijayanayaka, Consultant Dermatologist, Teaching Hospital, Karapitiya, to the head table.